Good morning, ladies and gentlemen. I am glad to be part of this conference, joining virtually to give my talk. First of all, I would like to thank the conference organizers for inviting me to give a talk in this international event. In this talk, I'm going to share my experiences in terms of developing the integrated pest management. Actually, when we talk about IPM, people think IPM is merely a package of practices to tackle the pest and diseases to reduce the use of chemical pesticides. But there are a lot of scientific research to go around to develop an IPM technology. And then it involves a lot of participatory research involving various stakeholders. So this morning, I'm going to share the science of developing and promoting the IPM across the different countries in Asia and Africa, which is a part of our ongoing program in World Vegetable Center in Taiwan. The science of developing and promoting integrated pest management in vegetable crops. So when we talk about vegetable crops, vegetables are actually high value crops. They can generate a huge amount of money per unit area when you compare with the cereal crops or other crops. So there are a number of pest and diseases infesting the vegetable crops in the tropical conditions, and they cause huge losses. And in order to avoid the losses and in order to maximize the income from the vegetable cultivation, the farmers resort to the use of chemical pesticides, which is actually the predominant means of the pest control in Asia as well as Africa. And this is a scenario I'm showing it from Cameroon in Africa. So most people think that the pesticide use is relatively less in African continent, but that's not true. So the vegetable crops receive a lot of chemical pesticides, and obviously this leads to a huge amount of adverse effects on the environmental health as well as the human health. So in order to reduce the adverse impacts of the chemical pesticide, historically, we have been advocating the use of integrated pest management approaches. Needless to explain the components of integrated pest management package, which is based on the foundation of the resistant varieties, but it's a combination of like biopesticides, biocontrol agents, pheromones, and various cultural practices, and also the need-based and the rational use of the chemical pesticides. And when you talk about the resistant cultivars, here comes the gene bank of World Vegetable Center. Actually, it is the world's largest public sector vegetable gene bank, having about 60,000 vegetable varieties, having about 439 species, covering 174 genera, collected from 151 countries or region. So because of the access to a huge amount of the genetic resources, we used to screen those germplasm from time to time to look for the resistant sources for various pests as well as the diseases. And here comes some of the examples. For example, we identified the tomato germplasm that are resistant to the whitefly, which is not only causing the direct damage, but also they transmit the begomavirus diseases in various crops, including tomato, causing almost 100% losses if the crop is left unprotected. And interestingly, some of the genetic material that are resistant to whitefly, they are also highly resistant to the two-spotted spider mite. And then these materials are also resistant to some of the lepidopteran caterpillars like Tuta absoluta, or currently it's called Thorimia absoluta, which is an invasive pest problem originated from South America and moved to Europe, and from there spread across Asia, Africa, and the Mediterranean region. And another example for the host plant resistance, we have screened a huge amount of onion genetic resources against the Thrips tabaci, which is onion thrips. Again, besides causing direct damage, it's also transmitting the Irish yellow spot virus, which is a tosphovirus disease, which is also causing a huge amount of loss. So we identified at least two resistant varieties, which are not only resistant to the thrips, but also they are producing a significant amount of yield because of the larger bulb size compared to the popular varieties cultivated among the farmers in East Africa, for example, like Bombay Red onion. And another example is the amaranth genetic resources, in which we have several species and hundreds of genetic materials. And obviously, the amaranth leaf webber is a major pest problem in both Asia and Africa. So we have identified a couple of resistant varieties for the amaranth leaf webber. And similarly, we have also identified the okra germplasm, which is having the aphid as a major problem in both Asia and Africa. So we have screened more than 700 different okra genotypes against aphid, the cotton aphid or melon aphid, Apis gossypi, and we identified several resistant varieties which have been introduced in African countries like Cameroon. So next to the insect-resistant varieties, we do carry out research on insect pheromones. One of our signature pheromone products is the eggplant fruit and shoot borer pheromone. So eggplant fruit and shoot borer is a major and significant pest problem in both Asia, primarily in Asian countries, including India, Bangladesh, and Southeast Asian countries. And we have developed an integrated pest management package, mainly based on the pheromone traps, but also including some of the cultural practices like healthy seedling production and the prompt removal and destruction of infested fruits and shoots, and withholding of the use of chemical pesticides in order to encourage the proliferation of natural enemies. And in fact, we, after an intensive research trials in South Asia, especially in Sri Lanka, Nepal, India, and Bangladesh, we were scaling out this IPM package in both Bangladesh and in India. Here, just I have highlighted the impact of this IPM package. You can see the huge difference between the IPM adapters and the non-adapters in terms of the use of chemical pesticides, in terms of quantity, and also the numbers. There is a significant reduction among the IPM adapters for the use of chemical pesticide, which resulted in significantly higher yield, but also it provided significantly higher income per unit area. So as of today, this is the scenario in Bangladesh. This picture was shot in Jessur, which is a major vegetable production hub in the southwestern parts of Bangladesh, where farmers continuously grow eggplant in stretches of area. Then you can see the installation of the pheromone traps in eggplant as well as other cucurbit crops to manage the critical insect pests like the eggplant fruit and shoot borer in eggplant and the fruit flies in cucurbit crops. And another example for the pheromones comes for the legume pod borer, which is called Maruca vitrata, which is a major pest problem on most of the vegetable legumes as well as the pulses. And obviously, 3 pheromone compounds have been identified in this particular insect. The pheromone blend has been developed using the major and minor components. And if you look at the performance of the pheromone lures, it's very interesting. For example, in West Africa, in countries like Benin and Ghana, a blend of 3 pheromone component is very effective, whereas in another country in the same region, Burkina Faso, only the major compound is attracting more number of insects. So there is a huge geographical differences. When we brought this pheromone to be tested in Asia, we did not find any attraction despite a huge amount of pest population flying in the field conditions. We did not find any attraction to these pheromone blends in countries like Taiwan, Thailand, India, Vietnam, or Cambodia. So we started focusing on why such a variation exists. And interestingly, if you look at this particular slide, there are three major and minor pheromone components. I used the different colors to differentiate them. And look at the blend ratio of the pest population in Taiwan. The major compound is 52%, and another compound is close to 46%, and the other minor compound is only 2.5%. If you look at the insect population in Thailand, in fact, the major compound is only about 37%, whereas another minor compound is actually 55%, and another minor compound is close to 8%. So there is an interesting differences among the populations of the same species, showing the variations in the pheromone compounds. So this is why we can expect a differential responses of the male moths to the pheromone blends in different geographies. So we went ahead to understand the species complex existing in this particular species. Then interestingly, we found at least the presence of three different putative species. One particularly present in Asian and African continent, and the other one is present in Oceania, including Australia and the Pacific Islands. The other one is present in the South American continent. So then we went ahead with the research. We found there is a synergism exists between the pheromones as well as the host plant volatiles. For example, the volatile from cowpea crop, one of the host plants of the legume pod borer, the major component of this host plant volatile is 1-octanol, and the 1-octanol along with the pheromones actually attracting more number of insects. Then besides the 6 pheromones which are commonly present in the beetles as well as the Lepidopteran moths, we also worked on the aggregation pheromone. One good example is the striped flea beetle. The flea beetle is a major pest problem on the brassicas, especially the leafy brassicas in Southeast Asia. However, in the recent years, it has also become a big problem in South Asian conditions, including India and Bangladesh. And these flea beetles are known to produce an aggregation pheromone, usually produced by the male sex, but it is attracting both male and female sex to congregate. So we were trying to identify a similar pheromone in the Philotritus triolata, which is the striped flea beetle. We identified a pheromone compound. And another interesting fact is this particular insect is having the myrosinase enzyme, which is commonly present in the plant species, especially in the brassicas, where the myrosinase enzyme breaks down the glucosinolate compound into the simple molecules. But interestingly, we found the flea beetle is also having this particular enzyme, so it is producing the glucosinolate compounds, which actually acts as a synergist along with the Philotrita pheromones. And we identified some of the additional compounds besides the Himachala 911 diene. There is another fraction of compounds present in the aggregation pheromone, which is actually increasing the attraction of the pheromone chemical. So additional research is necessary to develop an appropriate blend, which is more attractive for the flea beetle. It should be based on the aggregation pheromone compounds under the host plant volatiles. So next to the pheromones, another important area to be focused on is the biological control. So one of the most successful biocontrol program that we implemented from World Vegetable Center is the diamond-back moth biological control. I don't need to explain the importance of a diamond-back moth, which is one of the most serious and cosmopolitan pest problems on brassicas worldwide. So we introduced a larval parasitoid, which is called a Diadigma semiclassum, which is highly adapted to the highlands or the cool season conditions. When you look at the history of the Diadigma semiclassum, it originated from United Kingdom, and it was introduced in late 1930s into New Zealand to control the diamond-back moth on the forage brassicas. After hearing its success in New Zealand, this parasitoid was introduced into Indonesia in 1950s. However, it established and performed quite well after the introduction of the biopesticides like Bacillus thuringiensis in Indonesia in 1970s. So upon learning its success, we introduced this parasiticide into Taiwan in 1980s. After extensive research, after realizing its potential, we introduced this biocontrol agent into many countries in Southeast Asia in different projects, and subsequently in the late 90s into South Asian countries, including India. Then the beginning of this century, we also introduced this biocontrol agent into the Eastern and Southern African countries. And this was carried out by our lead entomologist, Dr. Talekar, who is well known for the diamond back moth biocontrol program. And in order to complement the performance of the larval parasitide, we also identified and introduced a pupal parasitide, because the larval parasitide may not control the diamond back moth 100%. So there will be some population becoming the pupa. So we introduced the pupal parasitide Diadromus scholaris, which complemented the performance of Diadema semiclassum. So both these candidates are highly adapting to the highland conditions. So we still have the problem of managing the diamond back moth in the lowland brassica production system. So there is one candidate called Cotesia pluteale. The current name is Cotesia vestalis, which is a larval parasitide, which is able to control the diamond back moth caterpillars in the lowland condition. But still, as I said, there could be some population of the caterpillars becoming pupa. So we were trying to look for a pupal parasitide, which is heat tolerant and adapted to the lowland brassica conditions. So we explored it in the Mediterranean countries, especially in the Syria, and we did a lot of explorations in Syria, and subsequently we were able to identify a heat tolerant parasitide collected from a place called Homs in central Syria. And this is the location where we collected this parasitide, and we did extensive research on their heat tolerance, and we confirmed it's a heat tolerant strain of Diadema semiclassum, which is an interesting finding. And then when we wanted to explore this parasitide and introduce it in the lowland conditions, we were not able to go back to Syria because of the civil war situation. But it's very interesting to note that there is a heat tolerant strain of parasitide is existing. And coming back to the biocontrol program of legume pod borer, obviously, when you look into the literature, there are a huge diversity of parasitides reported. But unfortunately, most of these parasitides generalist parasitides. So we started exploring for the species-specific parasitides of this particular insect in Southeast Asia, which is supposed to be the center of origin for this particular insect. So we did a lot of research. Then we were able to find some of the specific parasitides like Phenerotomys silepte, Therophilus maruca, and Therophilus javanus, which have been eventually introduced into the African continent, where there was no presence of any species-specific parasitide for legume pod borer. But this is a serious pest problem on cowpeas, which is the primary source of protein for the majority of African population. So these candidate parasitides have been introduced into many countries in West Africa, and they have been established quite well to perform in the field conditions. And coming to the biopesticides, I would like to highlight one example for the legume pod borer. Obviously, there are reports about the presence of cypovirus or the granulovirus for the legume pod borer from China or from West Africa like Benin, and from East Africa like Kenya. However, there was no report on the nucleopolyhedrose virus. However, back in 2005, we were able to isolate a nucleopolyhedrose virus from the dead cadavers of the Maruca vitrata, and we studied them extensively and made sure that they are very species specific, and we developed them eventually as a biopesticide formulation. And again, along with the parasitides, we introduced this virus formulation into West African countries for the management of the legume pod borer. And this particular NPV was found to be having a more synergistic activity when it is combined with the biological products like neem oil or the oil from the Jatropha. So this is also an effective biopesticide that have been developed in Asia and introduced into Africa. But besides the Maruca vitrata, there are also other caterpillar insects. So we were doing a lot of work on the efficacy of the Bacillus thuringiensis toxins, with the idea of choosing some Bacillus thuringiensis commercial formulations to be used along with the biocontrol agents and also the NPV formulations. So we found Cry 1AB, Cry 1CA, are quite toxic against the Maruca vitrata, and then this will give us the opportunity to choose two different formulations, because Cry 1A is the major toxin in Bacillus thuringiensis subspecies Kurstaki based formulations, and Cry 1CA is a major toxin in the Bacillus thuringiensis subspecies Izavai based formulations. So then we also did a lot of work on the entomopathogenic fungal formulations against the Maruca vitrata. So then we took all these biopesticide formulations and we tested them. Like we tested the individual biopesticides, like the different BT formulations, neem oil, entomopathogenic fungal formulations in different locations in Cambodia, for example. And then we tried to assess the efficacy of different biopesticides against the aphid. So here you can see some of the biopesticides are able to reduce the aphid infestation, and also they are able to reduce the infestation of the legume pod borer when you compare to the untreated controlled treat plots in different locations. So after identifying the most effective biopesticides, actually we put them together into an integrated pest management package and tested them again in different locations in different seasons. Here also you can find the black bar is the IPM package, and the gray bar is the farmer's practice of calendar-based application of chemical pesticides, whereas the white bar is the untreated control plots. So in all the locations, you can see the IPM package is able to reduce the aphid infestation in levels comparable to the chemical pesticides, or even they are able to perform much better than the chemical pesticides. So here we are able to reduce the pest infestation, but we are also able to substantially reduce the use of chemical pesticides compared to the farmer's practice. And the similar results have been also obtained for the pod borer infestation in different locations of Cambodia. So all these results have been published in different publications. If you are interested, you can refer these publications for additional details. So in summary, the development of IPM has a lot of scientific research component. So it's not an overnight job. It is not done in a few months. So the development, identification and development of resistant varieties, development of pheromone lures, and the identification of biocontrol agents, development of biopesticides, they all require intensive research and strong scientific evidence to be generated before putting them together into an IPM package. So I just highlighted some of the tip of the icebergs based on our research outcomes. So I just pulled out a few examples how we develop the different components of IPM and how we put them together into a package, and how we conduct the participatory IPM package validation in the farmer's research, the farmer's field conditions, and took the feedback from the farmer and then refine the IPM package before promoting them among the farmers. So here comes the clear role of the public as well as the private sector. For example, when we do a lot of discovery research like development of the resistant varieties, development of the pheromone lures, biocontrol agents, or the biopesticides, and then doing the research in the field conditions, they all require a lot of involvement of the public sector institutions like World Vegetable Center or the universities or the research institutions. But then we want to promote them among the farmers, where we need to transfer the technologies to the private sector, which have the capacity to develop or to produce the right kind of formulations, the right kind of production methods, and the right kind of technologies and logistics to produce the IPM technologies on the large scale, and then distribute those technologies to be available within the reach of the farmers at affordable prices. So here comes the strong partnership between the public as well as the private sector organizations in terms of discovery research, piloting research, and then the scaling of the IPM technologies to reduce the use of chemical pesticides and to enhance the sustainable production of safe vegetables. So with this, I would like to thank the organizers once again for giving me this opportunity. And if you have any questions, you can write to me in this email. Thank you very much.